Thank you everyone for attending the seminar today. I'm going to show you some work that I've been involved in since I was a postdoc at the US Army Institute for Surgical Research, where we were very much focused on the physiological responses to hemorrhagic stress, hemorrhage being one of the leading causes of death on the battlefield. And so we're trying to investigate how the body responds to that stress, particularly with a focus on the brain. So that's really where my main area of interest is for my research, is on brain blood flow and brain oxygen regulation under different stresses, but a focus on hemorrhage. So I'll be talking to you a little bit about the concept of brain blood flow and blood pressure variability and how that might impact the tolerance to a hemorrhagic stress, and then how perhaps we can develop interventions which might improve tolerance to that stress. And I'll be flipping between screens. I'm sure most of you have seen that happen before. So first of all, what is variability? So we know traditionally single static snapshot measurements of cardiovascular status have been assessed clinically. These include blood pressure, heart rate, respiration rate. So you go to a doctor's office, they take a blood pressure, that's your blood pressure, right? Well, we all know that's not correct. We know that blood pressure varies over the day. Blood pressure varies beat to beat. Blood pressure varies when you stand up, when you sneeze, when you go to the bathroom. All sorts of different things will influence blood pressure. This is just an example of the daily variation in blood pressure and heart rate. So you can see in the top panel we have blood pressure, and you can see that there's these variations in blood pressure across the day where you have a reduction in blood pressure when you're sleeping, and then it will increase when you wake up, but it will vary quite a lot throughout the day. The second line on the figure at the top here is the standard deviation. And so even within a single time of day, there is variance in your blood pressure within your own blood pressure responses, but also between individuals. So it varies over time of day, and it varies between individuals. And the same can be said for the heart rate responses as well on the bottom panel. So we know that blood pressure, heart rate, all cardiovascular variables are not static. They're very, very dynamic. And the modern monitoring technologies have allowed us to really explore that dynamic process with much more resolution than we've ever been able to explore before. So here's just one example of a blood pressure tracing. This is an individual in normovolemic status, so normal blood volume, and this is a continuous blood pressure recording over about a minute time. So you can see that obviously blood pressure will vary a little bit as you're just sitting here, but not too much. Systolic pressure here is 116/78, so that's kind of a normal blood pressure response. What about when you stress the system? This is an example of someone who's undergone a 50% reduction in central blood volume by application of lower body negative pressure. And you can see that the image of that blood pressure tracing is now quite different. You can see these fluctuations in the blood pressure response. However, if a blood pressure recording were to be taken with a traditional arm cuff around the brachial artery, your actual systolic and diastolic blood pressures would be identical. So if you're just taking these single snapshot measurements, it doesn't tell you a lot about the physiological status of this individual. But the pattern of that response might be much more informative. And that's really what I've focused on for a lot of my research in the past few years. So this is not a new concept. This is a figure from Guyton and Harris back in 1951, where they were recording blood pressure responses in hemorrhaging dogs. And you can see that the blood pressure response here, this is not just systolic and diastolic pressure, right? This is the ECG waveform, or the R wave spikes at the bottom here. These are actually, there's variations in blood pressure throughout these oscillations. And so they published on this back in 1951 that there were characteristics of the blood pressure tracing that we should really be focusing on. But as I say, the technology that we're able to utilize, particularly in human physiology experiments, has really allowed us to get into some of those dynamic responses that we once weren't aware of. So this figure is showing you kind of the different frequencies at which blood pressure and cerebral blood flow can vary. So the top panel here is blood pressure, the bottom is flow velocity using transcranial Doppler ultrasound. And you can see that there's different domains in which the blood pressure and blood flow are oscillating. So you've got this single beat here, okay? So this is at 1 beat per second if your heart rate's 60 beats per minute. And then as you compress that waveform, you can see that there's variations in pressure at very different time scales. And we can assess those different frequencies of variations using different mathematical techniques such as fast Fourier transform. And that's what you see on the right-hand side here, where you see these peaks occurring at different frequencies. Okay? So at the 1 hertz frequency right here, that's our cardiac frequency, so how often your heart is beating will be at a certain frequency, and we can measure that. We can also assess the high frequency here, which is generally related to respiratory frequency. So if you increase or decrease your respiration rate, you'll see a different spike occurring within that band. With this figure, everything's kind of washed out with this huge peak here at the ultra-low frequency, but if we cut that out, you would see a peak in the high-frequency band of respiration. And then we have low-frequency oscillations in pressure and flow. These are generally thought to be associated with sympathetic activity, which has about a 10-second cycle. And so you'll see bursting of sympathetic nerve activity in Dr. Fidel's lab and others. You'll see it occurring around about every 10 seconds. And then you have very low-frequency oscillations. These are generally thought to be myogenically mediated. And then on the lower end, we have much slower rhythms such as hormonal variations, circadian variations, basically everything that takes a long time. You'll see at the very, very low end of that frequency spectrum. So this is just to show you kind of we can get into some of the physiological mechanisms that are responsible for these variations in pressure and flow, and almost everything in the body will vary with these same timescales. So can anyone tell me what this oscillatory profile is? Anyone can guess? No? It's the faculty oscillatory profile when you're starting out as a new faculty member. So go from postdoc or job search to new assistant professor, lab not being ready, to having lab renovations complete, to lots of ideas but no personnel, first graduate student waiting for IB approval, data collection commences, hypothesis rejected, first paper submitted as senior author, revision, first paper accepted, writing grants, grant success, grants not discussed. If anyone's interested, over 5 years, this is the frequency, okay? 8 cycles over 5 years. so I sometimes joke I live in the frequency domain, okay? So this is the kind of concept I'm talking about. But there are oscillations in all sorts of frequencies, and this is just a joke, a little poke at that. So there are varying time scales of oscillations. So there's short term, so beat to beat or minute to minute variations. There's mid-term, so hour to hour or within 24 hours. And then longer term variations, so day to day, week to week, month to month, year to year variations. And so all of these can be important in terms of the underlying physiological mechanisms involved. So many of you have no doubt seen some figures such as this one. This is from Dr. Doug Seals, who was actually a speaker at the TACSM meeting last week, and he presented something similar to this. And this is what we often think about when we talk about blood pressure variability or pulsatility. Okay? In this case, we have a young vessel on the left-hand side and an old vessel on the right-hand side. Okay, and for the trainees in the room, what is the concept that we're talking about here when the vessel will not expand and accommodate the increased volume? Yeah? What else could it be called? Yeah? Does the term compliance ring any bells? Right? So basically the ability of that vessel to expand and accommodate that volume will have an impact on the pressure within that vessel. Okay, so if you have a very stiff vessel, then you won't get expansion of the vessel, so the increased volume will result in a high pressure within that vessel. Right? So here's an example of a young individual with the increase in stroke volume into the vessel. You'll get basically accommodation of that volume, and the difference in pulse within that vessel will be quite low, so low pulsatility effectively. Okay, when you get older, with a decrease in the compliance of that vessel, that same change in volume will result in a large increase in the pressure, or a large difference in the pressure. So it's a highly pulsatile waveform that you'll see. Okay, so with an increase in pulsatility, this can have an impact on end organ function, and particularly the vital organs of the heart, the brain, the kidneys, and even in the eye. Okay, so that increase in pulsatility is often what we think about when we hear the term blood pressure variability. We think about it in quite a negative context. And so there is a whole host of different papers that have been published in the literature describing this negative impact of high blood pressure variability on clinical outcome. It's associated with stroke, hypertension, kidney disease, a whole bunch of different, these are just one PubMed search on some of the papers that are associated with blood pressure variability and poor clinical outcome. However, I'd like to present to you the other side of the story. So is blood pressure variability a friend or is it a foe? So much of the research has focused on the negative clinical consequences of blood pressure and cerebral blood flow variability. But there are scenarios where increased blood pressure and cerebral blood flow variability may actually be protective or have some positive effect. And a colleague of mine, Shek Zheng over in New Zealand, he and I wrote a review on this a few years ago, basically describing these two parts of this story. And what I'd like to focus on for you today is the friend part. Okay, so what are the scenarios where an increase in variability may actually be protective? So we all know that adequate cerebral blood pressure and perfusion, so cerebral perfusion and oxygenation are essential to maintaining consciousness and for the survival of cerebral tissues. We know that reduced central blood volume or hypovolemia will challenge the cerebral perfusion, such as during standing, with dehydration, and during hemorrhage, and hemorrhage has really been the focus of the work I've been doing. Traditionally, the primary mechanism of this protection was a protection of absolute arterial pressure and absolute cerebral blood flow. And this has been in the literature for many, many years, that if you're able to maintain your blood pressure and maintain your cerebral blood flow, you'll be protected against syncope or from damage to the tissues. There's now accumulating evidence that suggests, however, that oscillatory pressure and blood flow may be protective under certain physiological or clinical conditions. And I'll just talk to you about those conditions today. So the stressor that I've been focused on is hemorrhage, as I mentioned, and I'm sure many of you are familiar with this box, the lower body negative pressure chamber, that I know is used in many of the labs here on the campus, and I've been using since my graduate work. So this is, you can probably recognise who this is in the photo. This is a subject exposed to a lower body negative pressure stress, and we can progressively decrease the pressure inside the chamber until the subject becomes pre-syncopal. So that's a point where they have symptomology such as lightheadedness, dizziness, nausea. Their systolic blood pressure will drop below 80 millimetres of mercury. So we're taking everybody to this classic endpoint of tolerance. As we apply that negative pressure, and this is just one profile that I've used quite often in our experiments, you'll get a progressive reduction in stroke volume as blood is pulled down into the lower body and away from the heart and away from the head. So effectively, you're decreasing the amount of volume coming back into the heart, you're decreasing the amount of volume being ejected out of the heart, and therefore the amount of volume being perfusing the brain. Okay. So this is our model of hemorrhage that I've been using for some time. So when I was a postdoc, we basically had the same protocol running for over, it was about 8 years in the lab, where we took, in this case, it was 135 subjects to the point of pre-syncope. So everyone did exactly the same study. They all came in, we measured the same parameters, and we were able to classify individuals as high tolerant or low tolerant. So of this group, we had 42 subjects who were low tolerant and 93 subjects who were high tolerant. And we based this on their ability to reach the end of this minus 60 millimetre of mercury lower body negative pressure. So if you made it to the end of that 5-minute step and you're able to move to the next step, you were high tolerant. If you could not finish that step, you were low tolerant. Okay, so this is how we dichotomize those two groups. So we use some classic techniques. Again, many of these are familiar to you. Transcranial Doppler ultrasound. We were measuring the velocity within the middle cerebral artery. So you can see the probe on the side of the head here. This is our waveform that we were able to collect velocity measures through that artery. And then we assessed all the other basic cardiovascular responses. So here we have stroke volume, and we can see that in both the high tolerant and the low tolerant subjects, there's progressive decreases in stroke volume. Okay, and all of these responses are reported up to minus 60 millimeters of mercury LBMP. This was the last common level of negative pressure that all subjects were able to tolerate, and then the high tolerant subjects went on to higher levels. So this is the last common level for most of the subjects that we were assessing. You can see that there is a slight protection of stroke volume in the high tolerant subjects, but it's really quite close as we progress down to these lower levels of LBMP. Then at pre-syncope, on the right-hand side, we have the high-tolerant subjects who were able to get their stroke volume down to almost 60% reduction from baseline, versus the low-tolerant subjects were only, if we progress, at about 40 to 45%. So the high-tolerant subjects were able to tolerate a greater reduction in central blood volume than the low-tolerant subjects.
Did they have the same stroke volumes of breath?
Yes, they did.
Absolute?
Yeah, absolutes, yeah. So we generally just report the relative change. This is from the phenometer, so I think most of us appreciate that it's a more accurate measure if it's relative versus absolute. But even absolutes at baseline are the same. Yeah. We don't measure things like volume, like blood volume status in these subjects, but that's an assumption that we make that it's the same. So we then assessed the arterial blood pressure response. This is mean arterial blood pressure. You can see the high tolerant subjects in the solid circles here had a protection of arterial pressure up to that minus 60 LBMP level, whereas the low tolerant subjects had a progressive fall in blood pressure, which was associated with their presyncopal status. At presyncope, however, their mean arterial pressure responses were the same. Okay, so the reduction in mean arterial pressure was the same whether you were high tolerant or low tolerant. When we had a look at the middle cerebral artery velocity, so this is the velocity through that major intracranial vessel, again we saw there were decreases in velocity in both groups. There was a slight protection, however, in the high tolerant groups compared to the low tolerant group at that last level, that last common level of LBMP. However, when we assess the presyncopal response, you can see that the high tolerant subjects in blue here had a lower absolute cerebral blood velocity than the low tolerant subjects. So the high tolerant subjects could tolerate greater reductions in central blood volume and also greater reductions in cerebral blood velocity. Okay? So what we were curious about, however, was when we examined the waveforms, we saw quite distinct differences between subjects who are high versus low tolerant. So in the top panel here, we have one representative subject who only made it to the second level of LBMP. Okay, that was a low tolerant subject. On the bottom panel is our one representative high tolerant subject who made it to about the fifth level of LBMP. And this is the cerebral blood velocity, so TCD velocity measurement. So you can see clearly that there are distinct differences in the waveform characteristics of those individuals. And this is the last 3 minutes prior to presyncope for both subjects. Okay? So what we were able to do is run our fast Fourier transform analysis and extract the frequencies at which these oscillations are occurring. So something interesting, though, just to point out here, is that the mean cerebral blood velocity for these two subjects were identical. Okay? So if we were just measuring mean cerebral blood velocity over this last 3 minutes of LBMP, there'd be no difference between the two. You couldn't distinguish a high tolerant from a low tolerant subject. But clearly, something's going on with the waveform characteristics that do differentiate these subjects. So we use this fast Fourier transform analysis. So just briefly, I won't go into the complicated math because I don't know it. I just run it on a software program. So this is arterial blood pressure that we have. This is the beat-to-beat measurement from our phenometer. We then extract out the mean arterial blood pressure from that recording. And then running our fast Fourier transform analysis, we're able to assess the different frequencies at which that blood pressure is oscillating. And in this case, we have a low frequency range of 0.04 to 0.15 hertz. And as I mentioned before, this is generally considered to be sympathetically mediated at about that 10-second cycle. Or if you have a subject who's breathing very slowly, you'll also see a peak within that range. So if you're breathing at 9 breaths per minute or less, you'll see a peak occurring at that low frequency range. And then the high frequency, as I mentioned before, as long as subjects are breathing at 9 breaths per minute or above, then you'll see a peak within that high frequency range. And so here's just some examples of an individual who has high oscillations, and then you run the fast Fourier transform and you see that peak occurring there at around about 0.12 hertz. And then an individual with low oscillations and effectively not a lot of activity going on there. So there's not a lot of power within any of those frequency bands. So when we went back and analysed the data for mean arterial pressure, now at the bottom of the page here, you can see that the mean arterial pressure low frequency oscillations are increasing at this -60 level of LBMP in the high tolerance subjects only. And there's a small increase in the low tolerance subjects, but it's really flattening out there at the end. And then at pre-syncope, there's a higher amplitude of oscillations in the high tolerance subjects compared to the low tolerance subjects.
Carol,
Yeah.
are they breathing on their own, or are you controlling their respiratory?
In this study, it was spontaneous breathing, and because we had so many subjects, there were some subjects who were breathing in the low frequency range, but the majority were breathing in the high frequency range. So they were all at least 9 breaths sort of or above. So, but it's a great question because it can totally interfere with the calculations and interpretation of the measurement. So when we assess the oscillations in mean cerebral artery velocity, you can see the same pattern here. The oscillations are increasing at that -60 level for the high tolerance subjects only and not in the low tolerance subjects at all. And then at pre-syncope, there were higher oscillations in the high tolerance subjects compared to the low tolerance subjects. So that was interesting. It was an interesting observation. We didn't know quite what to make of it and how it might be associated with increased tolerance. And we thought that we may be an anomaly publishing this, and we don't know what it means and may never know what it means. And then a study came out 2 years later that showed basically the same type of thing. So in this study, this is from Sam Lucas and Phil Ainsley, who I know a lot of you already are aware of. In this study, what they actually did was induce oscillations by pacing the breathing of the subjects. Okay? So in our subjects, we allowed them to breathe spontaneously, and we just assessed what we call the endogenous oscillations in their blood pressure and cerebral blood flow. What these investigators did was they actually had subjects in 2 conditions. One, they spontaneously breathed. In another, they forced them to breathe at 6 breaths per minute, so at that 0.1 hertz frequency. So here you can see on the left-hand side you have the spontaneous breathing condition. On the right-hand side, the controlled breathing condition. And you can see here we have blood pressure on the top. We have middle cerebral artery velocity, end-tidal carbon dioxide as a measure of arterial partial pressure of carbon dioxide. And then we have heart rate on the bottom panels. And you can see that they had subjects in the tilted position, and then they added on -30 millimeters of mercury LVMP. So it was quite a significant cardiovascular stress. And they wanted to get everybody to pre-syncope by tilting them up first and then adding on the lower body negative pressure stress. So you can see as we move from the spontaneous breathing here, you can see that the oscillations are increasing in the tilt plus -30 in pressure and velocity. But if you look at the controlled breathing condition, you can see that the oscillations are quite pronounced in both pressure and velocity, and they get even more pronounced when you add on the -30 millimeters of mercury LBMP. And as I said, they took all of their subjects to a point of pre-syncope. In this figure, on the left-hand side, you'll see the frequency domain spectrums. Basically, they're showing you in the solid lines, this is the controlled breathing condition. So at head-up tilt alone on the left, and at tolerance on the right, and these are all the parameters that they're measuring. And you can see those peaks are appearing in the controlled breathing condition. The amplitude of those peaks are much higher than during the spontaneous breathing condition. And interestingly, the time to pre-syncope was increased in the controlled breathing condition. So when subjects were breathing at 6 breaths per minute, or at 0.1 hertz oscillations, their tolerance to the head-up tilt plus LVMP increased. Okay? But when they assessed the middle cerebral artery velocity responses, the change from baseline to pre-syncope was identical between conditions. So this was similar to what we had seen in our lower body negative pressure experiments, where there was not a big difference in the cerebral blood flow responses between high and low tolerance subjects, but their tolerance obviously was different. Okay, so this was supporting what we were seeing, but now with a situation where they were forcing the oscillations at 0.1 hertz. We still don't know what it means, but it was confirmatory in a different model compared to what we had seen. So what is the mechanism for the oscillation? So we've talked a lot about respiration, and yes, low-frequency oscillations will occur if respiratory rates are low. The majority of subjects are, however, breathing outside the low-frequency range in our LBMP tolerance study. Respiration will mediate the high-frequency oscillations if your breathing rate is above 9 breaths per minute. And LF oscillations will occur even with paced breathing protocols. So if you're forcing those oscillations at above 0.1 hertz, you'll see this increase in low-frequency power under conditions such as lower body negative pressure, head-up tilt, or dehydration. Are they sympathetically mediated? So we know from many studies in the past that there is some mediation of those low-frequency oscillations through the sympathetic nervous system. And with increases in absolute sympathetic nerve activity during LBMP stress, the muscle sympathetic nerve activity and arterial blood pressure oscillations increase together. And that data is presented here from my postdoc lab with Bill Cook. And you can see here we have muscle sympathetic nerve activity on the left-hand side, muscle sympathetic nerve activity, low-frequency power on the right-hand side, and then on the x-axis we have LBMP as a percentage of their maximum. So similar to what you do for a VO2 max test, we just split up all of the data into 20% increments and plotted that here for our... We have 20 subjects who made it all the way to presyncope with the sympathetic nerve activity recordings. So you can see that sympathetic nerve activity increases progressively during LBMP, and the low-frequency power in MSNA also increases. And you do get a little drop-off here at the end. We're not sure what that means, but you basically see this linear association between oscillations in sympathetic activity and absolute sympathetic activity. When you then plot the sympathetic low-frequency oscillations versus diastolic pressure, low-frequency oscillations, there's a very tight relationship between these two. So as you increase the oscillations in sympathetic activity, obviously you'll increase the oscillations that you see in blood pressure. And just as another visual, this is actually a recording from our lab where we measure sympathetic activity in the radial nerve. And you can see on the top is the blood pressure recording. In the bottom we have the sympathetic bursts here. Obviously every time blood pressure falls, you get bursting of the sympathetic nerve, and that will result in a recovery of blood pressure. So there's tight coupling between the oscillations in pressure and the oscillations in sympathetic activity. So this is true for not only diastolic pressure but also for systolic pressure and mean arterial pressure. This is just an example showing you the relationship between MSNA LF and pressure LF. Again, that very tight relationship. And this is with our N of 20 subjects. We were able to dichotomize those subjects into low tolerant and high tolerance. So we had 5 subjects who are low tolerant, 15 who are high tolerant. So we have a smaller N on the low tolerant side. But what you can see here is the black circles represent the high tolerant subjects, the open circles are the low tolerant subjects. And I've only put a line through the relationship that has a low p-value here. So you can see in the high tolerant subjects, this relationship's quite tight. In the low tolerant subjects, it's much lower. Okay? So this relationship between pressure and sympathetic activity, low frequency oscillations, is really being held by the high tolerant subjects. And that relationship is much, much weaker in the low tolerant subjects. When we do our traditional coherence analysis, so this is using our transfer function analysis, and effectively in the frequency domain, we're looking at the correlation between oscillations in pressure and sympathetic activity. You can see that high tolerant subjects have an increase in coherence between pressure and MSNA, but low tolerant subjects really have no change across LBMP. And I appreciate the error bars on the low tolerant subjects are huge because they have a very low N, but even so, there are some differences here at the higher end of LBMP. So there's a difference in the coupling between pressure and sympathetic activity in individuals who can tolerate this central hypovolemic stress that we don't see in the low tolerant subjects. So I don't know what that means yet, but it's an interesting observation. So what we can say, and also here we have the correlation, the connection between pressure oscillations and cerebral blood velocity oscillations. So again, in our N of 20, we have this tight relationship, and then when you split it out into the high and low tolerance subjects, again, the high tolerance subjects maintain this tight relationship. The low tolerance subjects are really not changing much at all. Okay, so there's not a real association between oscillations in pressure and flow in those low tolerance subjects. So we know that high tolerance subjects exhibit a linear association between low frequency oscillations in MAP and sympathetic activity, MAP and mean cerebral blood velocity. And these may be modulated by sympathetic nervous system, so that the oscillations we observe in the cerebral blood velocity may be modulated by sympathetic activity, but we don't know if that is direct, as in, is there sympathetic activity in the brain that's oscillating, which is inducing the oscillations in cerebral blood velocity, or is it purely a passive response to the changes in blood pressure variability? So that's kind of an unknown right now. So here's just one of the flow diagrams we have in terms of some of the mechanisms of protection that we're trying to explore. So we have observed that an increase in low frequency oscillations in arterial pressure is associated with tolerance to central hypovolemia. I won't say it's a cause and effect at all yet, but there is an association. This could be due to either an increase in sympathetic nerve activity or an increase in the coupling between pressure and sympathetic activity, which may lead to protection of mean arterial pressure, which is what we saw in our high tolerance subjects versus low tolerance subjects during LBMP. A protection of mean arterial pressure will lead to increased cerebral perfusion pressure. So remember that cerebral perfusion pressure is mean arterial pressure minus intracranial pressure. Okay, so the protection of arterial pressure should protect cerebral perfusion pressure, which in turn should increase cerebral blood flow. On the other side, if we have an increase in low frequency oscillations in arterial pressure, this seems to be associated with an increase in low frequency oscillations in cerebral blood flow, which could increase shear stress within the vasculature, which could increase vasodilators that are released, which may increase vasodilation of the cerebral vessels and increase cerebral blood flow. So I know that one of the questions you might be asking is, well, how are you seeing increases in cerebral blood flow when you saw no protection of cerebral blood flow? Well, it may be a limitation in the technique that we're using right now. So as most of you know, using transcranial Doppler ultrasound is limited because we're only measuring velocity and not flow within the vessel. So if this is correct in that we're seeing a dilation of the vasculature, then we may get an increase in flow, which is masked by the measure of velocity. Okay, so this is really pointing us toward measuring something else other than just velocity within the intracranial vessels, and I'll get to that in a moment. Also, with that increased low frequency oscillations in cerebral blood flow, perhaps there's a modified pattern of oxygen delivery. Okay, so maybe this modified pattern of oxygen delivery is facilitating the delivery of oxygen, which is meeting metabolic demand, which is stabilizing oxygen extraction. And if we have an increase in cerebral blood flow, which is increasing delivery, then that may also facilitate the delivery of appropriate oxygen to the tissue. So basically, supply is meeting demand. Okay, so a lot of these are just hypotheses right now that we're trying to explore. And the key ones that we're interested in is the sympathetic modulation that's going on, the increase in shear stress, the release of vasodilators, the vasodilation of the vessels and subsequent increases in flow, and also the role on oxygen extraction. So this leads me to the last study that I'll describe, and it's one that we've just been starting. We've just finished our phase 1 portion in the lab, and I have a group of students and trainees over here from the lab. So Garen, this is your study. So basically what we are doing with this study is, again, we're using lower body negative pressure. We're exposing subjects to different lower body negative pressure profiles. The first one we do is we call it the 0 Hertz profile. So we pull the chamber pressure down to minus 60 millimeters of mercury, and then we keep it there for about 10 minutes, and we see how the subjects respond. We also then have two different oscillatory profiles. So this is our 0.1 Hertz profile, so we pull the chamber pressure down to minus 60, and then we oscillate between minus 30 and minus 90 for 5 seconds at each pressure. And so we're inducing that 10-second cycle in assessing how the subjects respond to that stress. And then finally, we have a 0.05 hertz oscillation. So we pull the chamber pressure down to minus 60, and then we oscillate the chamber every 10 seconds between minus 30 and minus 90. So the average pressure is the same in the chamber. So they're all hypovolemic, but one condition or two conditions we're oscillating the chamber at different pressures to try and force those oscillations in pressure and cerebral blood flow. And this next slide shows you two examples of the oscillations, and this is in middle cerebral artery velocity, 0.05 on the top, 0.1 on the bottom. And when we run the fast Fourier transform analysis, you can see that we're getting quite nice peaks at those different frequencies. So the grey line here is 0.05 hertz. Okay, so this is the 0.05 hertz oscillations. The dashed line here is 0.1 hertz. So that's what we're looking at right here. And then the baseline is the black line here. So this is just the minus 60 static profile. Okay, so just to demonstrate that we're inducing these oscillations as we had designed. So here are some of the, we're right now just measuring the middle cerebral artery velocity and the posterior cerebral artery velocity. So we're interested in the regional perfusion of the brain. So both the anterior portion with the MCA and the posterior portion feeding the brainstem with the PCA. In the future, we're also trying to investigate the extracranial vessel flows. So we can get cross-sectional area of the internal carotid artery and the vertebral artery to actually measure inflow into the brain rather than just relying on these velocity measurements. We don't have the data for this right now. That's phase 2 for Garen's PhD project. So I'll just be showing you the phase 1 data with the TCD today. We also use near-infrared spectroscopy to measure cerebral oxygen saturation of the frontal lobe. So here we have a probe that's on the frontal lobe. You can see the photograph here with the probe right here on the front of the forehead. This technique, we make a lot of assumptions. It's a non-invasive technique to measure oxygen saturation of the cerebral tissue. Basically, the sample volume, we take it as 75% venous, 20% arterial, 5% capillary, so it's a mixed venous sample. It's not just arterial, it's not just venous, it's not just capillary, it's a mixed sample. So basically the way that we interpret this is obviously oxygenated hemoglobin is coming through the arterial side of the circulation. We get oxygen extraction into the cerebral tissues at the level of the capillary beds, and then on the venous side of the circulation, we'll get a increase in deoxygenated hemoglobin and a decrease in oxygenated hemoglobin. And from that, we can calculate oxygen saturation of the tissue by dividing the oxygenated and deoxygenated into the oxygenated hemoglobin. And so if we have a decrease in oxygen saturation, we interpret this as an increase in oxygen extraction, because we have an increase in deoxygenated, a decrease in oxygenated hemoglobin. So from that, we get a SCO2 value. If it decreases, this equates to an increase in extraction. Right. We measure, right now we're measuring sympathetic activity just with circulating catecholamines and just with norepinephrine so far. But the next study will also be measuring sympathetic nerve activity in the radial nerve. And as I say, this is the same picture I showed you before, showing you that the nice, close or tight association between pressure and sympathetic nerve activity. We don't have these data yet because that is also phase 2. Okay, so here's the data that's of most interest to us. So here we have the tolerance to the 0 hertz profile and the 0.1 hertz profile. And you can see that in all but one outlier, well, they're not an outlier, we don't know why they're decreasing tolerance, but all other subjects had an increase in tolerance to the 0.1 hertz oscillation. So most subjects were able to tolerate the entire protocol when the chamber pressure was oscillating compared to when it was just static at minus 60. Similarly, if we assess the 0.05 hertz oscillations, then most subjects were able to tolerate that 0.05 hertz profile compared to the 0 hertz. So we're seeing increases in tolerance for both of those oscillatory profiles, which we weren't sure if we were expecting, but it's interesting data. So these are the hemodynamic responses. Oops, gone too far. So interestingly, in the 0 hertz profile on the left-hand side, we have greater reductions in stroke volume compared to the two oscillatory profiles. So at 0.1 hertz versus 0.05 hertz, the reduction in stroke volume here was attenuated compared to the 0 Hertz profile. Okay? This then resulted in an increased heart rate response in the 0 Hertz compared to those two oscillatory profiles, which subsequently resulted in similar reductions in cardiac output and similar reductions in mean arterial pressure, regardless of the profile. Okay? So the cardiac output and pressure were not different between groups, despite differences in stroke volume. And that's likely due to the compensatory increase in heart rate here, for sure. So that's our basic hemodynamics. Sympathetic activity, as you may expect, the norepinephrine was higher in the 0 Hertz profile compared to the oscillatory profiles, so greater sympathetic activation with that greater reduction in central blood volume, so it makes some sense. When we assess the intracranial velocity and oxygenation responses, we saw something quite interesting here as well. So this is the MCA velocity responses. So the decrease in MCA velocity was identical between the different profiles. We don't know, however, whether, again, we're masking the effect of flow because we're measuring just velocity and not diameters and not flows. So we're not sure yet. Hopefully with our extracranial vessel measures of flow, we'll get more clarity on that question. Interestingly, with our posterior cerebral artery velocity measurements, in our 0.05 Hertz profile, there was a protection of cerebral blood velocity. I'll just point you to the N of 5 at the bottom here, so our numbers are small. But this is consistent with what we've seen in the past, where individuals with higher tolerance have a protection of posterior artery velocity compared to those with low tolerance, which may be associated with perfusion of the brainstem, where autonomic activity is controlled. So this kind of makes sense to us. And just in terms of numbers, the ratio of subjects who are able to tolerate the 0.05 was greater than the 0.01. So more subjects were able to tolerate the entire 0.05 profile compared to the 0.01. So this is in agreement with what we've seen previously. When we assessed the SCO2, the oxygen saturation of the frontal lobe, we saw also that there was a attenuated reduction in SCO2 in the oscillatory profiles compared to the static profile, which, as I mentioned before, suggests that there was a increased extraction in the 0 Hertz profile compared to the oscillatory profiles, despite the fact that delivery, as with our measure of MCA velocity, was the same. So we don't know what this means either, but maybe there's the difference in delivery of oxygen is improving the efficiency or the extraction for meeting metabolic demand. Maybe metabolic demand is different within the brain, which is then influencing this response. So there, again, some questions that we're trying to explore. We have a measurement of a vasodilator nitric oxide. This is just a systemic measure from the venous blood. We measure nitrite, which is a metabolite of nitric oxide. There's no differences between the three conditions. However, if we observe the individual responses here between the profiles, you can see it's a mixed bag. We have 7 subjects, 4 of them have increases, 3 of them have decreases. So we need to increase our numbers for this analysis to see if there's anything with this particular measurement. We're also measuring a prostaglandin metabolite to see if that might be elevated as well. So trying to get at the vasodilator release with these increases in oscillations. So again, there's lots of unknowns. We're starting to explore the sympathetic component, the shear stress, vasodilator, and flow responses is all really in phase 2 of this study. But we've got some interesting data that may be suggestive of some responses that we're exploring. And then the oxygen extraction side as well is what we'll continue to assess. I think right now one of the limitations is that we're measuring systemic release of these vasodilators. Ultimately, I'd really like to be able to measure over the brain release of these factors, so measuring jugular venous versus arterial, to see if there's something going on locally within the brain rather than just these systemic measures. So many of you might be asking, well, how would this ever be applied to a hemorrhaging scenario? You're not going to be carrying around a lower body negative pressure box in the back of an ambulance. So I've actually been working with my colleagues down at San Antonio at the US Army Institute of Surgical Research, where I did my postdoc work, where they have decades of expertise in actual hemorrhage using a rat model predominantly. And they can hemorrhage their animals 40 to 55%. And what we've actually started to do is use these tiny little Hookinson cuffs around the thighs, and we can inflate and deflate the cuffs at certain frequencies. So the mechanism might be different, but we can induce oscillations in pressure. You can see at the bottom here, these are mean arterial pressure oscillations by inflating and deflating those cuffs around the thighs of these rats. So we are able to explore much more invasive measures with the animal model. Obviously, we can assess things like AVO2 differences across different circulations. We can assess vital organ damage in the brain, heart and kidneys, and also these circulating agents as well, both locally and systemically. So these are some studies that we're hoping to get up and running as soon as I can hit a grant. So, but they have been running some pilot work for us using this technique. So just in summary, there is evidence that indicates short-term variability in cerebral blood flow may be protective under conditions of reduced flow, and this could be applied to situations such as hemorrhage and stroke. There may be, the harmful versus protective effect of variability may be time dependent. So short-term variability in the acute setting may be protective, but mid to long-term variability, so years and decades of variability, may be harmful to the vital organs. There is a potential application of pulsatile perfusion therapy under varying conditions of reduced blood flow, and this is just a list of potential applications in the clinical setting. And this also may facilitate permissive hypotension, which basically is allowing an individual who has lost a lot of blood volume to maintain their pressure at below what would be considered normal, and then actually induce these oscillations to protect vital organ perfusion. And in the battlefield, out in one of the applied environments, reducing fluid requirements is a big deal. So not having to carry around so much fluid for medics to treat soldiers is an important issue. So that's one of the potential applications on the military side. So just in closing, I'd like to acknowledge all of the individuals who've been associated with the work I presented today, my trainees in the lab over at UNT Health Science Centre, my friends and colleagues at the US Army Institute of Surgical Research, Bill Cook, who was also at the Army Institute when I started. He was then at UTSA and now is at Michigan Tech. And the funding from the Department of Defense, the American Heart Association, and seed grant funding from the Health Science Centre. So thank you, everybody. Take any questions.